everyone. We will perform review and exercise for lab exam number 1. This lab exam number 1 will be over lab number 1, number 2, 3, 4, 5, 6, 7, 8, and 9. There are 9 lab experiments will be used for lab exam number 1. So for the best result, make sure you repeat lab experiment 1 until 9, as many as possible. So do not worry, okay? You will get the highest grade. Okay, you don't have to worry that if you take it and it's going to be lower grade. No. So your grades will be only the highest score that you get from this lab experiment. So repeat your lab experiment several times in order to know, to learn what kind of questions will be on the lab exam number 1. And again, prepare some piece of paper or and pencil to write your answer. There will be about 2 or 3 seconds after each question to write your answer before the answer key is given and explained. And after finish the exercise, check your answer. How many are correct and how many are incorrect. Repeat the process until you get 100% correct answer, and make sure that the 100 correct answer are based on your knowledge. So therefore, make sure you repeat the lab experiment before taking this exercise. And it is very likely that you will get a very, very good score if you perform this review and exercise correctly and follow the instruction. Okay, so we start with number 1. The purpose of lab safety rules and protocols is to A, B, C, or D. Choose your answer. Okay, so lab safety rule is for protecting everyone in the lab, okay, from injury and maybe infection. So that's the primary purpose of lab safety rules. Which of the following food is allowed in the laboratory? Which one? A, B, C, or D? The answer is never. Okay, never. So no food or drinks are allowed in lab. You have to put it outside the lab. Which of the following beverages is allowed in the laboratory? Choose your answer, A, B, C, or D. Okay, again, never. Okay, no food and drinks are allowed in the lab. This is for your own protection. Okay, there may be infectious disease or pathogen that maybe go to your food or drink. So make sure you don't bring any food or drink to the lab. Lab coats, gloves, face masks, and goggles are examples of A, B, C, or D. Okay, so it is lab equipment is maybe, okay, maybe supplies by your instructor, or maybe it's provided for you. But the main function of this, okay, equipment is for personal protective equipment, or PPE. Okay, so they are PPE. To protect your skin and clothing from hazards such as chemicals and infectious agents, the blank should be worn all the time in the lab. A, B, C, or D. Okay, so it should be lab coat. So you have to wear lab coat all the time inside the lab. Which of the following personal items provide the same protection as goggles? A, B, C, or D? So eyeglasses, soft lenses, sunglasses, or none of the above? Of course, none of the above. Okay, so your eyeglasses cannot replace the goggles. So you have to wear goggles when performing some activity like dissection. When you are required to wear closed-toed shoes in the lab? When? A, B, C, D. Okay, the answer should be always. So always wearing the shoes inside the lab. No sandal. Okay, no open toes inside the lab. Soap should be applied to blank hands. A, B, C, or D. Okay, of course, you have to wet your hand before applying the soap. So it should be on the wet hands. How long should hands be lathered while washing? A, B, C, or D? 10 seconds, 20 seconds, 10 minutes, 20 minutes? It is at least 20 seconds. So usually people say you can sing happy birthday. Okay? Happy birthday to you. It's about 20 seconds when you wash your hands. Disinfectant is used on work surface. A, B, C, or D? Okay, make sure if you work with pathogens, then the disinfectant has to be used before and after. Okay, so all of them. So before, after lab finish, and also when there is a spill. So all of these choices. Okay, before start the lab, after finish the lab, and also if there is spill on the bench or desk. Glass slides, scalpels, razor blades, cover glasses, and capillary tubes are classified as hazardous, infectious, sharp, or biohazard. Choose your answers. So all of them is sharp. Okay? They are sharp object. Glass slides, pipettes, and capillary tubes should be disposed in regular trash, recyclable trash container, sharp container, or biohazard bag. Yeah, because they are sharp, then it has to be put in sharp container. So every lab will have this biohazard, sharp container, and regular trash container. So put them in sharp container. Paper towel, lab notes, and lens paper should be disposed in A, B, C, or D. All these items are regular trash, so you can put them in the regular trash container. When you are finished with this dissection tray, scalpel, and forceps used in dissection, you should blank. Put them in sharp container, put them in biohazard, or put them in regular trash, or wash them in the sink. Okay, so all of these are reusable. We use it again, so do not throw them away. So make sure you wash them in the sinks and put them on the desk or on the bench. Okay, wash them. When you are finished with dissecting preserved specimen, you should blank. Put them in sharp container, put them in biohazard bag, put them in regular trash container, or wash them in the sink. Okay, so the specimen is going to be thrown away, but you have to put them in biohazard bags. So that should be the answer. Which of the following should be done first in the event of lab accidents? Call 911, B, panic, notify campus security, or notify the instructor. The first thing you have to do is notify the instructor. Then the instructor will do the next process, like, for example, call 911 or notify campus security. The group that receive variables during the experimentation is called control group, experimental group, constant group, or normal group. Okay, so they receive variables. They become the experimental group because they receive some things during experimentation. The group that does not receive variable during the experimentation is called control, experimental, constant, or normal. Of course, the one that does not receive the experimentations or variables is called the control group. Which of the following statement is true about hormones? A, B, C, or D? Okay, just remember, hormone produced by endocrine gland. Okay, produced by the endocrine gland. Then these hormones mostly will be carried by blood. Okay, blood circulation. So look for that one. They are produced by endocrine gland and circulated in the blood circulation. So this should be the answer. Which of the following statement is true about the target cell? Okay, hormone produced by the glands, okay, then carried by the blood. It released into the target cell. And the requirement to be a target cell is that target cell have the receptor for that hormone. That's the requirement for target cell. So look for that statement or that signs. Look at it very close. No, the receptor has specific receptor. So this one over here, right? So this is the answer. So they have receptor for the hormones. A hormone that targets the nearby cells without entering the circulation is called A, B, C, or D. Okay, so D will be not the answer because exocrine is not for hormone. Okay, it is for other fluid like tears, sweat, mucus. So this is not the answer. So maybe autocrine, endocrine, and paracrine. So this is the difference. Okay, so you have glands that produce the hormones. Okay, produce the hormones. Okay, if these hormones go to the circulations, blood circulation, and the target cell is here, okay, it has a receptor. Okay, this hormone over here that go to the circulation is called the endocrine. Okay, if the hormones produced by the glands, okay, and go to the neighbor cell that has receptor for that hormone, okay, this hormone is called paracrine. Okay, if the gland produce hormones, and this is the gland produce hormones, and actually this gland also have receptor for the hormone, so the hormone is used by itself. This hormone is called the auto. Auto means self. Okay, so autocrine. Okay, so this is nearby cell, nearby target cell. So which one is going to be? A hormone that targets the nearby cells without entering circulation is called the paracrine. A hormone that is sensed by its own cell that produces it's called the, we already mentioned that, this is auto. Okay, so the gland is actually has a receptor for the hormone, so it's used by itself. This is autocrine. A hormone that must circulate in the circulation to reach the target cell is called the, of course, endocrine. So we already explained that. Compared to the neurotransmitters, hormone action is faster, slower, about the same. They cannot be compared. Okay. So neurotransmitter is released by neurons, which is our nervous system, and the action is really, really fast. Okay, so fast action. But hormone is produced by the endocrine gland. It's produced slower action. So it's gonna be slower compared to neurotransmitter. Which of the following hormones increase body's metabolic rates and temperature? Okay. Choose your answer. There are many hormones produced by our body, and one of them that have receptors on almost all of our cells to increase metabolism is the thyroid hormone. So the answer is thyroid hormone. Remember, insulin is for increase, decrease glucose, right? So this is not the answer. Calcitonin is used for decrease calcium in the blood. Okay. Growth hormone is to increase growth, okay, which is bone and muscle. So this is not the answer. So the answer will be thyroid hormone. Which of the following hormone is secreted in response to decreased body temperature? Okay, A, B, C, or D. So when our body temperature decrease, okay, so body temperature decrease, we need to increase it. Okay, so increase our body temperature. How? By increasing metabolic rate. Okay, when our cell active, it will produce heat. That heat will increase our body temperature. So which hormone increase metabolic rate? Thyroid hormone. If thyroid hormone is not released, usually your body will be blank to regulate body temperature. A, sweating, sleeping, shivering, or dying. Now, if thyroid hormone is not produced, okay, so our body temperature will be down, will be decreased. So in order to increase it, then what happens? Our muscles will be shivering. So will be shivering during cold weather. It is what? It is to increase the body temperature. So shivering is the answer. Body shivering to control body temperature requires a greater use of A, B, C, or D. Okay, choose your answer. Okay, remember the easiest way to increase body temperature is by releasing thyroid hormone. So thyroid hormones, when it is released, will increase body temperature. However, during some emergency, we maybe do not produce enough thyroid hormones. We increase body temperature by shivering. And shivering is muscle contraction. And this is require a lot of oxygen. So therefore, this shivering will require a greater use of oxygen. Administration of PTU, profile thiouracils, during experiment, so this is actually part of our lab experiments, would produce effect of what? A, B, C, or D. So PTU will decrease thyroid hormone production. Okay. So when the thyroid hormone production decrease, then your body will need other way to increase body temperature, will be shivering. So in the experiment, the one that received PTU, they produce less. This is actually rat, so using rat during the experiment. Rat that received PTU, PTU, the thyroid hormone will be decreased, then this rat will perform this shivering. You will see it later. And shiverings can be measured by the use of oxygen. Okay, so rat that receive PTU will use more oxygen during the experimentation. Okay, now this is the question for that. The experimental groups that receive administration of PTU, profile thiouracil, will have A, B, C, or D. Okay, so the rat group that receive PTU, remember their thyroid hormone will be decreased, right? So they need to perform shivering of their muscle. And the shiverings will require a lot of oxygen. Okay, so therefore you can measure it by, they will show the higher oxygen consumption. So they are going to use more oxygen compared to the normal group or control groups. Respirometer for PTU experimentation is used for measuring blank consumption in the mice. Okay, so choose your answer. We are talking about using, shivering, which is shiverings, muscle contraction, and muscle contraction require oxygen. So the equipment, the respirometer, measure the oxygen consumption. Which of the following hormones decrease blood glucose? Okay, so in the experiments, we use fish, and then we measure the hormones. And what hormone decrease glucose in the blood? And this is very famous hormones, of course, the insulin. Which of the following statements about insulin is true? A, B, C, or D? Okay, so insulin decrease glucose in the blood. So just look for that one. Increase is not correct. Decrease glucose level in the cell is not correct. Okay. Increase absorption of glucose by liver, muscle, and fat. Okay, and decrease the absorption of glucose. So it's going to be increased absorption of glucose from the blood, by liver, muscle, and fat. So when the liver, muscle, and fat absorb the glucose from the blood, then the glucose level in the blood will be decreased. Okay, so this is the answer. Decrease the glucose level in the blood by absorption of glucose by liver, muscle, and fat cell. So this is actually the target for insulin. Okay, the target cell for insulin is actually liver cell, muscle cell, and fat cell. So they will absorb more glucose from the blood, and then it will decrease glucose level in the blood. In which of the following condition fish will swim with slow and lethargic movement? Normoglycemia, hypoglycemia, or hyperglycemia? Choose your answer. Okay, normoglycemia means glucose level is normal. Okay, in the blood of the fish is normal. Hypo means low glucose levels in the blood. Hyper means high glucose level, okay, in the blood. Now, which one do you think will cause this fish to swim really slow and weak or lethargic? So when we don't have enough glucose in the blood, then we have become lethargic, okay, really weak. So this should be hypoglycemia condition. Okay. Now, number 35. In which following condition fish will swim very fast and darting movement is very fast movement? Normal, hypo, or hyper? This is the same thing with kid. Kid, when they take a lot of sugar, what happens? They are hyperactive. So this is hyperglycemia because of too much glucose in the blood. In which of the following condition fish will swim with regular and smooth movements? A, B, C. Of course, this is the only choice. Normal condition will give regular, normal, smooth movement, or swim. To measure hematocrit, blood sample must be A, B, C, or D. Okay, these experiments, okay, in the lab experiments, to measure the hematocrit, you have to put the blood in the centrifuge tube, and then you centrifuge them. When you centrifuge the blood, it will separate the blood into two main or three main separation. The bottom one is going to be the hematocrit, the red blood cell. Okay, the top one will be plasma. And between the middle, there is a buffy coat, which is mostly white blood cell and platelets over there. Okay, red blood cell, which is the hematocrit. This is hematocrit, mostly normally 45%. And the plasma, 55%. The rest, less than 1% over here, will be the buffy coat, white blood cell and platelets. Okay, what we have to do in order to get or to measure hematocrits, we have to centrifuge the blood. After centrifugations, the blood sample will produce how many layers of blood part? Okay, so after centrifugations, again, there will be 3 layers. Okay, the lower is hematocrit, which is red blood cell. The middle is white blood cell and platelets. Okay, and the top layer will be the liquid or blood plasma. So 3 layers. After centrifugation, the blood sample will produce, okay, I think it's the same questions. 3 layers. When a blood sample centrifuges, the top layer represents what? A, B, C, or D. And the top layer will be plasma. Because this is the lightest one. So the lightest components will be on the top. The heaviest components will be at the bottom, which is red blood cell. So the top one is the plasma, and normally 55%. When the blood sample centrifuges, the buffy coat in the middle layer represents, and remember this is going to be the white blood cell and the platelet. It is less than 1%. When the blood sample centrifuges, the bottom layer represents blank. Okay, we already explained this. This is going to be the red blood cell, and sometimes it is also known as the hematocrit, and normally is 45%. The most abundant form elements in the blood is the, remember form element, there are 3 types. Red blood cell, platelets, and white blood cell. Red blood cell is about 99% of the form element. The rest, platelet and white blood cell, is about 1%. So the most abundant one will be the red blood cell. And the scientific name for red blood cell is erythrocyte. Okay, so the answer will be erythrocyte. Leukocyte means white blood cell. Thrombocyte is platelets. Okay, so erythrocyte, red blood cell. So that's the answer. Hematocrit is a measurement of the percentage of blank in the blood. Okay, so we already mentioned this. Hematocrit is the same as the percentage of red blood cells in the blood. And red blood cell is this erythrocyte. Hematocrits of blank would be about normal. Okay. We talked several times that hematocrits normal percentage is around 45%. Oxygen transport in the blood is provided by the blank. So which one carry oxygens? Of course, it's going to be the cell. So which cell? It is the red blood cell, which is the erythrocyte. Inside the red blood cell, there is a protein is called hemoglobin, okay, or HB, that will bind the oxygen. So hemoglobin inside the red blood cell. So the answer will be red blood cell. If a person has hematocrit lower than 38%, he might be has abnormal blood condition called A, B, C, or D. So less than 38%. So normally, okay, the HT should be 45%. So this is red blood cell. So what does red blood cell do? Carry oxygen. Someone that have less oxygen level is called anemia. Okay, so anemia is lower oxygen level or lower red blood cell level or lower hematocrit. So it's lower than 45 is anemia. A person that has higher than 60%, he might be has abnormal blood condition called A, B, C, or D. Of course, it is not anemia. We already know that anemia is lower than 45. So this is very high, above 60%. Okay, so leukopenia, it is not. Penia mean low. Leuco mean white blood cell. So this is not the answer. Leukocytosis, cytosis mean high. Leuco mean white blood cell, which is not the answer. Okay, the answer will be polycythemia. That's the only answer over there. Which type of white blood cell is this? So this is under the microscope during your lab experiments. So this is white blood cell. There are 5 types of white blood cell, okay, based on the abundancy. Remember, never let monkey eat banana. Okay, neutrophil, lymphocyte, monocyte, eosinophil, and basophil. Neutrophil has a nucleus with many lobes. So it's going to be look like this, many lobes. Okay. Lymphocyte, it is the smallest type of white blood cell with really big nucleus like this. Okay. Monocyte, this is the biggest white blood cell with the nucleus look like a kidney. Okay, so kidney-shaped nucleus. That's monocyte. Okay, remember that. Eosinophil. Okay, it has nucleus look like earphones. Okay, 2 lobes, and it has red granules. Okay, red granule. Eosinophil. Basophil. You don't see the nucleus because what it has blue granules, and blue color is also the color of nucleus. So therefore, the nucleus is covered by this blue granule. So you don't see the nucleus on basophil. Okay, so if you look at this different structure of white blood cell, then you see this one, it has many nucleus. One nucleus with many lobes. Okay, so it's going to be the first one over here, the neutrophil. Okay, so this is how neutrophil look like under microscope. Next one. Look at this one. What is this? Remember, it has red granule. And the nucleus is look like earphones over here. So what one is that? It's going to be eosinophil. Look at this one. You don't see the nucleus, right? It's all covered by these blue granules. Which one is this? This is the basophil. Look at this one. The size is almost similar with the red blood cell. Okay, so this is actually red blood cell. Yeah, red blood cell. So this is the smallest white blood cell with a very big nucleus. Okay, so what is this? This is the lymphocyte. Next, look at this one. This is really big white blood cell with kidney-shaped nucleus. So which one is this? This is the monocyte. So make sure you memorize the shape and the structure of each white blood cell: neutrophil, eosinophil, basophil, lymphocyte, or monocyte. Okay, the most abundant white blood cells are, okay, don't forget the sequence, never let monkey eat banana. Okay, the most abundant one will be neutrophil. Okay, and this is the one that fight against bacteria. Lymphocyte fight against viruses or infected cell by virus. This is also bacteria, the monocyte. Eosinophils is parasites. Okay. Basophil, this is mostly allergens, okay, allergic reaction and inflammation. Both of these sometimes causing the inflammations. Eosinophil and basophil. Okay. Monocyte neutrophil, they are also known as phagocyte. Okay, cell that eat bacteria or foreign cell. So the answer, the most abundant one will be neutrophil. The least abundant white blood cells are, we already mentioned that, never let monkey eat banana. So banana or basophils are the least abundant type of white blood cell. The white blood cell that has greatest nuclear variations is the A, B, C, D, or E. So this is going to be the one that has many lobes. Sometimes it's look like this. Sometimes just look like this. Okay. Sometimes like that. So which one is this that has many lobes? This is the neutrophil. The white blood cell that has reddish stain granule is, choose your answer. We remember eosin actually mean red, so this is the one that carry red granules, the eosinophil. The white blood cell that has bluish stains granules, and usually these granules will cover the nucleus. You don't see the nucleus. Is which one? Baso. So baso means blue. So basophil is carry blue granules. Okay, so all of three of these, okay, all of these called the granulocyte. They carry granules. Eosinophil carry red granules. Basophil carry blue granules. Neutrophil carry kind of neutral, clear granules. Lymphocyte, monocyte is called the agranulocyte. They don't carry granules. So the cell is really clear. There is no granule. So like monocyte, for example, is going to be carry the kidney-shaped nucleus without any granules. Okay. The lymphocyte is going to be small with big nucleus. Okay. Neutrophil with these many lobes and the nucleus and really clear granules. Eosinophil look like earphones. Nucleus with red, bigger granule. And then basophil will have blue granules, really big, that cover the nucleus. Okay, so these are the granulocyte. The other two over here, agranulocyte, clear plasma. Okay, clear cytoplasm. What does a differential white blood cell count? Okay, A, B, C, or D. Okay, so this is differential white blood cell. It means that you actually count all type of white blood cell. Okay, different type of white blood cell. So it is not the total number. Okay, it is not the total number. So it is going to be percentage of different type. Different type. So which one? Different type of white blood cell in the fraction of the whole white blood cell type. So different white blood cell in the whole white blood cell. So this is to be the answer. Not the percentage of white blood cell to the whole blood. Okay. What is the most important first step in differential white blood cell count? Finding the basophil, recognizing each type of white blood cell, counting each type of white blood cell found under the microscope, or making a good blood smear or specimen. Okay, of course, finding basophil is not really important because basophil is very rare. Sometimes you don't find it. You don't have to really find the basophil during the experiment because it's very rare. Recognizing is important, but it is not the first step. And counting each type is also not the first step. The first step is making a very good blood smear or specimen to be checked under the microscope. That should be the first step. And in the lab experiment with this differential white blood cell count, the very first step, very important, is how to make a very good specimen, blood smear. The antigens related to ABO blood groups are located where? A, B, C, or D. Remember, for ABO blood group, the antigens are located on the red blood cell. So there are antigen A. If the red blood cell carry antigen A only, so blood type gonna be A blood type. If the red blood cell carry the B antigens, so this is B antigen, this is A antigen, then the blood type is going to be B. If red blood cell carry both A antigens and B antigens, then this person has AB blood type. If the red blood cell do not carry any antigen, this person has O blood type. So it is located on the red blood cell. The antibodies related to ABO group, blood groups are located A, B, C, or D. Okay, remember antigens, it is located in the red blood cell. On the red blood cell, this is the antigen A. So this person will be A blood type. Okay, so when someone is A blood type, which is carry antigen A, then he will produce antibody B. Antibody B in his blood plasma. So this antibody is located in the plasma. The surface of red blood cells of a person with type B blood has A, B, C, or D. So this is how to identify people with blood type. If type B, it means that he has antigen B only. That's it. It has B antigens on the red blood cell. The surface of red blood cell of a person with type AB blood has A and B. So both A and B antigen on the red blood cell. The surface of red blood cells of a person with type A blood has what antigens? So type A is mean only carry A antigens. So on their red blood cell. The surface of red blood cell of a person with type O blood has anti. Oh, this is the surface of red blood cells. It's going to be, I think it's a wrong question. Okay, it is the answer is antibody. Okay, so it is not on the surface. Okay, but in the plasma. In the plasma of a person with type O blood has what? So because he is O, okay, so O blood type do not have any antigen, right, on the red blood cell. So this is O. Therefore, he can produce both antigen and antibody A and also produce antibody B. Okay, so person with type O will produce both anti-A and anti-B antibodies. Okay, so yeah, so this because we changed the question. Okay, so the answer should be C. Okay, so talking about antibody. Okay, so this is antigens over here. Okay, it's not antibody. So this is one of the questions that you have to know what's wrong with this. Okay, when it is talking about on the red blood cell, then it is antigen. What? But when it is talking about antibody, it should be in plasma. Okay. In the plasma of a person with type A blood has, okay, so this is in the plasma, it means that it will be antibody. So this is type A. So type A means he carry antigen A. Okay, because he carry antigen A, then he will not produce antibody A. He will produce antibody B. So look for B. It's gonna be this one. In the plasma of a person with type B blood has, okay, again in the plasma it's going to be antibody. So just remember, someone with B type, okay, on the red blood cell, he will carry the antigens B. Okay, so that's make his blood is B. But because he has antigen B, then he will not produce antibody B, he will produce antibody A. Okay, look for A, antibody A. In the plasma, so again, this is going to be answer is antibodies. People with AB type, okay, what type of antibodies will be in his plasma? So again, for AB person, he carry both antigen A and antigens B. Okay? Because he carry antigen A and antigen B, then he will not produce antibody A, and also he will not produce antibody B. So it's going to be neither. Okay, so neither anti-A or anti-B antibody. So he will not produce antibodies. In the plasma, okay, in the plasma, a person with type O, right? We already answered this one from the previous question that is incorrectly put on the screen. So O, blood type, no antigen. So no antigen. What does that mean? It means that he can produce antibody A and also antibody B. So both anti-A and anti-B antibodies will be in his plasma. If the blood from an individual with type A blood mixed with the antiserum A, anti-A, okay? So this is during the lab activity. You mix blood with antiserum in the first well, and then with antiserum B in the second well. What would be the result? So the blood type is A. Okay, so you put 2 wells over here. Okay, and blood type A. Okay, it means that he carry antigen A, right? Antigen A. So you put his blood over here and also here on the well A and well B. And in the well A, you put antiserum A, which is antibody A. Okay, and the B, you put antibody B. Okay, so what happened? Because he carry this antigen A, then on the well A, this blood will be agglutinated, will be clumping. Okay, clump. You can see it clearly. Over here, because he has antigen A with antibody B, is nothing. It's just smooth. Okay, nothing happened. Okay, so someone that have blood type A during the experiment, and the well A, his blood will be clumping. Okay, so the first well will show agglutination, the second well will not. So this should be the answer. Okay, if the blood from individual with B blood type mixed with antibody A in the first well, and then antibody B in the second well, what would be the result? And I know this is B, only the second well that will be clumping or agglutination. Okay? So the first well will not show agglutination, the second well will. So this should be the answer. If a blood from an individual with AB, okay, blood mixed with a antiserum A in first well and antibody B in the second well, what happened? Remember, A, B carry both antigens. And these antigens and the first well will be agglutinated with the A antibody and also will agglutinate with B antibody. So both well will be showing the agglutination. So the answer should be C. Okay, so both the the first and the second well will show the agglutination. If the blood from an individual with type O mixed with antibody A, antibody B, on the first and second well, what happened? Remember, O, no antigen. This means that there will be nothing happening. So the blood will be smooth, no agglutination. So neither the first well and second well will show the agglutination. That's how you type your blood. That's how you know your blood type, by using this antiserum A and antiserum B during experiment. The antigen related to rhesus blood groups are located. It's going to be the same. So the antigens located on the red blood cell. So maybe the antigen look like this. This is the antigen RH. Okay, so on the red blood cell. The A, B, and RH antigens are located, and again, all of them will be located in the red blood cell. Okay, so maybe antigen A, antigen B, antigen RH, they're all located in the red blood cell. The anti-A, anti-B, and RH antibodies are, okay, so all antibody produced in the plasma. Okay, they are dissolved in plasma. Which type of blood has all type of antigens on red blood cell and no antibodies in the plasma? Of course, if someone carry all type of antigen, there will be no antibody. Okay, so this person will carry all the antigens. So his blood's gonna be, so this is A, oh, sorry, this is A, this is B, this is positive or RH. So his blood will be AB positive. Which type of blood has A and RH antigens on red blood cell and anti-B? So this person carry antigen A and antigen RH, which is positive, right? And he will produce antibody B, okay, in the plasma. So this person will be what? A plus. Okay, just look the antigen. That's how you define the blood type, A plus. Which type of blood has B and RH antigen and produce, of course, antibody A? So it has B antigen and positive or RH antigen. So his blood will be what? Will be B positive. He will produce antibody A. Which type of blood has B and no RH antigens? And red blood on red blood cell and anti-A and anti rhesus antibodies in the plasma. So he carry an antigens B, okay, no RH, okay, and he produce antibody A and also antibody RH. Okay, so his blood is gonna be negative, okay, because no RH over here. So B negative. Which type of blood has no antigens on red blood cell and all type of antibodies in the plasma? So this person do not have any antigen on the red blood cell. Therefore, he will produce all the antibody. Okay, antibody A, antibody B, and antibody RH. Okay, so he doesn't have antigen. What blood type over here? So for ABO, he is O because no antigen, and for rhesus, he is negative. Right? So O negative. Which type of blood has B antigen and no RH antigen? Okay, it has B antigens and negative on RH. Okay, no antigen RH, and he will be B negative. Okay, that's easy to identify someone with blood type. Just look at the antigens. Okay, just look at the antigen. What antigen he has. Okay, now look at this picture. This is the real experiments. Okay, so this one put the antibody A. Okay, and this use okay, the antibody RH is put in this well. Okay, and then this B over here, we put antibody B. Okay, look at this one. When you put antibody A in his blood, there is nothing happening. It's smooth. Okay, so it means that no antigen A. So his blood does not carry antigen A. Look at this one. This is agglutinate, clump. Okay, so clumping or agglutinations, it means that this blood has RH antigen. So this blood is positive. Okay, look at this. It's also agglutinate. And so it means that he has B antigen. Antigen B. Now you combine this with this. Okay? So this is going to be his blood type. What is that? B positive. Okay, that's how you know blood type. Okay, John Doe has AB blood type. So AB blood type, it has antigen A and antigen B, has an accident and is required to have fast blood transfusion. The hospital does not have AB blood type, okay? But it has A, B, and O blood type. Which blood type should be used for the transfusion? Okay, you have to remember O is the universal donor. He can give blood to any blood type. So the answer should be O. But the best actually AB. AB need AB. But if in the emergency situations, O blood type can be used for any other blood type. Jimmy Neutron has AB blood type. Okay, which blood type should he receive for transfusion? Of course, I mentioned before that AB should be used first. Okay, but in emergency, the type O can be used. So this is the answer. Use the same blood type first. That's the rule. Okay? If not, then in emergency, O can be used. Which of the following artery is the most common use for palpation? Okay, so this is during experiment also, when you palpated your friend's artery, which one that you should use? It is on your wrist, okay? It is on his wrist, which is called the radial artery. Which of the following arteries can be palpated on the surface of the body to measure pulse rate? Okay, which one? Vertebral, no, you cannot use vertebrae. This is inside, it is in your vertebrae. Renal is inside your body, you cannot use it, okay? Common iliac is also inside your body, okay? So the one that may be used is the dorsal pedis artery, okay? It is on the foot, okay? This is your foot, you can use this dorsal pedis artery. Which of the following arteries can be palpated in the inguinal regions to measure pulse rate? Okay, so for the groin area, the femoral, which is on the thigh, the artery on the thigh can be used for palpation to measure the pulse rate. What do you measure when determining blood pressure? So blood pressure measure what? Of course, it's not vein, because vein, there is no pressure in the vein, okay? Arterial, this is not, because this is in the heart, okay? Capillary is also not, because it is really, the pressure is really low. The one that has higher pressure is arteries, okay? Whether aorta or artery. This is the one that we measure for blood pressure, arterial blood pressure. Which of the following arteries is used for determining blood pressure? Common carotid, femoral, radial, or brachial? So when you measure the blood pressure, okay, you using the sphygmomanometer, where you put it? On the brachial artery, on the upper arm. Okay, so this is brachial, it's called brachial area. So this is brachial artery. The stethoscope, so stethoscope is placed over blank when taking blood pressure. So we already mentioned that the sphygmomanometer with the stethoscope will be placed on the brachial artery to measure blood pressure. During ventricle contraction, the arterial pressure reach the maximum. So this is ventricle contractions, and it will give what? The systolic pressure, usually 120 normally, or less. When the ventricle relax, so this is ventricle relaxation, the arterial pressure still give number, drop to the lowest one. And it is called what? It is the diastole, usually, normally 80, okay, 80 millimeter per mercury. Diastolic pressure. Blood pressure is expressed as a fraction. The top number of this fraction indicates the top person. Usually, normally we receive 120 over 80, right? The top one is systolic. The bottom one, diastolic. Okay, so this is systolic pressure. Blood pressure is expressed in fraction. The bottom number, this fraction indicate, we already mentioned that, this is diastolic. Okay, 120 over 80. So this 80 is diastolic pressure. The first carrot cough sound occurs when the blood pressure is equal to the, okay, this is when we measure the blood pressure with that sphygmomanometer, we hear the sound. The first sound is actually the highest sound, okay, which is showing us the highest pressure. So what is that going to be? So 120, 80. This is the highest sound is coming from here, which is the systolic pressure. The last carrot cough sound occurs when the blood pressure is equal to the, and this is again, this is the lowest pressure, will be the measuring the diastolic pressure or coming from the diastolic pressure sound. Which of the following resting blood pressure would be considered normal? Okay, normal blood pressure. We always, we see this many times. This is now kind of a normal blood pressure, 120 over 80, right? So look for that one, 120 over 80 is normal blood pressure. Which of the following resting blood pressure, remember we take it in during resting time, not after exercise, okay, not after sleeping, but it's sitting, resting blood pressure, would be considered as hypertension. Yes, hyper means higher, okay, so higher blood pressure. 90 is really low, 110 is low, 120 is kind of normal, this is a normal blood pressure. 140, this is considered as hypertension, 140 over 90. Why did pulse rates decrease 5 minutes after exercise? So this is during lab exercise, okay, lab experiment. You ask someone to do exercise and you measure the pulse rate after exercise. Why it is increasing after exercise? Okay? A, B, C, or D. The reason to increase is because the tissue need more oxygen. Okay? Therefore, your blood pressure will be increased and the pulse rate telling you. So 5 times, 5 minutes after exercise, the pulse rate will be increased. The pulse rate decrease 5 minutes after. Okay, so decrease after exercise. So during exercise, the pulse rate will be higher. And then 5 minutes after that, it will be decreased. Why? Because after exercise, 5 minutes after exercise, your oxygen demand also will be decreased. So the pulse rate will be decreased. The blood pressure will be also decreased. When a person moves from sitting position to laying position, lying down, his blood pressure decrease. Why? Okay. The reason is because the blood does not have to travel against gravity. Okay, so the blood pressure will be decreased when we are laying down because it doesn't have to move against the gravity. Now, what happens? So this is during the experiment. When a person moves from sitting position to a standing position, his blood pressure increase. Why? Okay, sometimes when you stand up after sitting or laying down, your blood pressure suddenly increase. So why? Because there are more skeletal muscle will be active and need more oxygen. Okay, so the blood pressure will be increased when you're standing up. How many chambers does the heart consist of? Okay, we already have this in the lecture. So there are 4 chambers, right? 2 atria on the top and 2 ventricles at the bottom. So 4 chambers. The top chamber of the heart are called A, B, C, D. So the top chamber is atria. Okay, right atrium. If only one is called atrium, more than one is atria. Okay, so right atrium and left atrium. The bottom chambers of the heart is called ventricles. Okay, right ventricle and left ventricles. That should be easy, right? Which chambers receive fresh, oxygenated, or rich blood from the lungs? Right, left atrium, or right, left ventricle? Remember, the top chamber, which is the atrium, is the one that receives blood. Okay, so atrium, the top chamber, is the one that receives. So your choice between only right or left atrium. The one that receives rich blood from the lungs is the left one. Remember, left area of the heart is the one that have rich blood. Okay, because left atrium just receive blood from the lung, which is a lot of oxygen. Okay, so it's going to be left atrium. Which chamber receive deoxygenated or poor blood from our body tissue? Okay, so remember, it's going to be atrium. Okay, so the right atrium, the one that receive 3 veins from our body. Okay, superior vena cava, inferior vena cava, and coronary sinus. All of them carry poor blood. Okay, so it's gonna be enter into right atrium. Okay, just remember the right side of our heart carry poor blood. Which chamber pumps deoxygenated poor blood through the pulmonary trunk to the lungs? Okay, now we're talking about pumping of the heart, which is ventricle. And we already mentioned that the right side is the one that has poor blood. So it's going to be coming from right ventricle. Okay, right ventricle will pump the blood through the pulmonary trunk into the lungs. Okay, so right ventricle. Which chamber pumps the oxygenated rich blood through the aorta to the whole body? Again, the pumping of the heart is the ventricle. Okay, the left one is the one that pumps the rich blood to the whole body through this aorta. Okay, remember again, the left side has rich blood. So it's going to be from the left ventricle. Which is the function of 4 heart valves? A, B, C, or D? Okay, the valve is to make sure that the blood flow in only one direction. Okay, so this valve actually to make sure the blood is only going one direction. Okay, from the heart, go to the artery, go to the tissue, going back to the heart. Okay, so the valves in the heart will make sure that blood flowing in one direction only. It's not going back or mixed with the other blood. Okay, the tricuspid valve control the blood flow from or between blank and blank. Remember again, tricuspid, you have R over here, so it's gonna be on the right side. Okay, so between right atrium and right ventricle. So that tricuspid, right atrium and right ventricle. The bicuspid or mitral valve control the blood flow from between blank and blank. So bicuspid is going to be the other side between left atrium and left ventricles. Okay, so on the other side, left atrium, left ventricle, there is bicuspid or mitral valve. The pulmonary valve control the blood flow between blank and blank. Okay, the name is telling you, so it's gonna be from the right ventricle with the pulmonary trunk. There will be valve over here. It's called a pulmonary valve. Okay, so control between right ventricle and pulmonary trunk. The aortic valve control the blood flow between blank and blank. Okay, it's gonna be from the left ventricle, go to the aorta, and there is valve over here between left ventricle and aorta. Is called the aortic valve. Between left ventricle and aorta. What is the pacemaker of the heart? It is located on the right atrium. It's called the sinoatrial node. So right atrium, sinoatrial node or SA node. When the heart contracts, this due to electrical chains called A, B, C, or D. So these electrical chains, the boom and then increase boom like that. Okay, so this P, Q, R, S, and then T wave, all of them because of the depolarization. Okay, so increase of electrical change is called the depolarization. When the heart relaxes, this is due to electrical change called. So when it is relaxed, okay, the opposite of depolarization is called the repolarization. Okay, relax because of repolarization. What does an EKG, electrocardiogram, measure? Depolarization, repolarization, cardiac cycles, or all of them? Yes, it's measure all of them. So measure the depolarization, repolarization, and also the cycle, systole and diastole. Usually cycle take about 0.8 millisecond at normal cycle. And this is what the EKG measure. Okay, which letter indicate P wave? Okay, so P wave is depolarization of atrium, which is the first wave over here, the A. Okay, so A is the P wave. Now, how about the T? Which one indicate the T wave in the EKG graph? It's going to be the last one, T, which is E letter on that question. Which letter indicates a QRS wave? QRS. Okay, so it's going to be B letters on that graph question. Which letter indicate the QRS? Okay. Things should be B. The EKG components correspond to the depolarization of atrium. Again, this is how the graph boom start with the P wave, QRS, and then T. Okay, P, QRS, and T. So P is during atrial contraction. QRS is during ventricle contraction or depolarization, and T is during ventricle relaxations. Okay, so P is atrial. Atrial depolarization is going to be P wave. Which EKG component corresponds to the depolarization of ventricles? Okay, ventricles gonna be the highest one over here, the QRS. This is when the ventricle depolarize or contracts. So QRS. Which EKG component corresponds to the repolarization of ventricle? So this is relax. So relaxations measured by the T wave. Okay, this is repolarization. What can we get from dividing 60 seconds by the time between the two consecutive R wave? Okay, let's say we have this, and then we have another one, this. Okay, so this is P, T, QRS. Okay, P, QRS, and T. So this one cycle over here, if we divide it by 60, then it will give us the heart rate. Okay, so the heart rate given by dividing the 2R cycle or rate, yeah, and then with 60 seconds, so you have beats per minute. Okay, the heart rate. Which of the following can be used to calculate the heart rate? Okay, we already mentioned from R to R interval and divided by 60, you're gonna have the heart rate, and the matrix gonna be beats per minute. Okay, I think that's all for this review and exercise. Good luck with your first lab exam. Bye-bye now.